Organic chemistry 2. So let's get this over with, right? Okay. So I imagine that what we're going to go over today you've had before in other classes, so that's also kind of a nice perk about the last lesson, in that we're going to do a real broad overview of amino acids and proteins. So without further ado, let's just go ahead and get into it. So the reason why we're going to talk about these is because now we know about nitrogen in organic compounds, particularly the functional group amine. And there's this important type of molecule in biology called an amino acid. And so now it's time to talk about it. So they contain both an amino functional group, and that happens to be acting as a base, as well as a carboxylic acid functional group, which of course, as the name implies, behaves like an acid. And one of the reasons why this is so important is because with those two functional groups, you can connect amino acids together to form a polymer. So they can be connected together as polymers. Terminology is flexible, but according to the textbook anyway, if it has fewer than 50 units, we call that a peptide. And if it's bigger than that, then it's a protein. However, there's always gray areas. And a protein actually usually a protein. And again, I'm not an expert in this, but from my little bit of exposure to it, it seems like what we call proteins are usually multiple amino acid chains working together. Okay, it's not all one big long molecule. Like hemoglobin. Hemoglobin is actually 4 separate amino acid chains that come together. Okay, so what is a protein then? Well, a protein is it's a larger chain of amino acids. And the other thing about a protein, if we're going to call it a protein, is it has some sort of function, biological function. All right. So in nature, there are about 20 amino acids that are common. Okay, there are many other amino acids. Strictly speaking, any organic molecule that has both an amine functional group and a carboxylic acid functional group could be called an amino acid. But in nature and in proteins, we typically see about 20 of them involved. And all of them are what we call alpha amino acids because the amine group is on the alpha carbon. Remember that the carbon that's adjacent to the carbonyl, in this case, the carbonyl of a carboxylic acid functional group, the carbon that's adjacent to that, the alpha carbon, that's the one that also is attached to the amine functional group, okay? They're both connected to the same carbon then, effectively. The carboxylic acid and the amine are both connected to the same carbon. All right, so you got that diagram right there. So what distinguishes one amino acid from another? I mentioned earlier there's 20 of them that are typically involved in proteins. So what's different about them? If they have that one thing in common, that both the carboxylic acid group and the amine functional group are attached to the same carbon, well, how do they differ? Well, they differ in this thing called the side chain. In that diagram, it's just labeled as an R group, but it could be many things. Okay? All right, so let's talk a little bit about the structure of amino acids, including the variations in that R group. And spoiler alert, I'm not asking you to memorize I don't have them memorized, so I'm certainly not going to ask you to memorize those 20 amino acids that are found in nature. Don't worry about that. I'll leave that for biochemistry or some other biology course, molecular or something like that. Okay, but one thing I do want you to know about them is at physiological pH, that they really don't exist, purely speaking, as a carboxylic acid group and an amine functional group, because the acid behaves like an acid and the base behaves like a base, and so there's a proton transfer from the carboxylic acid to the amine, okay, at pHs that we are, accustomed to in living systems. And so we call that, there's a special name given to that, it's called a zwitterion, okay? And what it is, it's dipolar, so it's still neutral overall, but you've transferred that H plus over to the nitrogen, so that gets the formal positive charge, and you're left over with a carboxylate, which has a negative charge. So it still overall is neutral, but it's a polar situation where one end of the molecule is going to be positively charged and the other end is going to be negatively charged. Okay, and so the other consequence of that, and this is going to draw back from something we talked about way back in Chem 2, is it means that amino acids are this big fancy word, amphiprotic. Now that means, remember, that it can behave as both an acid and a base, whichever one is appropriate. Okay, so in an acidic solution, what happens is you end up with an overall positively charged product. Because in an acidic solution, that carboxylate takes the H plus from the hydronium. You know, remember that in an acidic solution, what that means is you have an abundance of hydronium. So the carboxylate acts as a base, and then your product overall has a positive charge now. Here it was neutral. To begin, it was neutral, right? One positive, one negative, they cancel each other out. But in the product, now, when it behaves as a base in an acidic solution, you get an overall positively charged product. And then just the opposite occurs in a basic solution. Remember that in a basic solution, hydroxide ions are the abundant one. And so they're just going to take away this extra proton off the amine. Okay, and so you get overall a negatively charged product. But the idea is that the same molecule behaves in different ways depending upon what conditions it's in, acidic conditions or basic conditions. Okay. So now let's just mention one thing about those side chains. Again, I don't expect you to memorize all of them, but know that they're in 3 categories. Neutral, and that's most of them. That R group does not participate in any acid-base reactions at all, for 15 of them. But then 2 of them are acidic, and then 3 of them are basic. Okay, also, just this is more trivia than anything else, humans can synthesize 11 of the 20, but the other must be obtained from food. Okay, I guess you could also, you know, pills, you know, dietary supplements. Sorry. Okay. Okay, so now there's this other, and it's kind of obscured on the screen, but you can see it there in your handout, that there's this other important point at which the amino acid is overall neutral. Okay? And that is this place where we call, I got to move it out of the way. Yeah, the isoelectric point. Okay? So that's where it's overall neutral. Isoelectric means, you know, it doesn't, no change in electrical charge is kind of what that word implies. And so it's balanced between the positive and the negative. But as I said before in the previous slide, if you are below the isoelectric point, if you're below that in pH, what that means is you're in acidic conditions that are going to impart to your amino acid a positive charge, overall positive charge. If you are above the isoelectric point for the amino acid, then overall you're going to have an amino acid that has a negative charge because it's, you know, and it's not, and the isoelectric point for each amino acid is different. Okay, so we're not talking about 7, where 7 is neutral. Because of the structure of each amino acid, each one has an isoelectric point that's different than 7. And so if you're at pHs below that isoelectric point, then you have a positively charged amino acid overall. If you are at pHs that are above the isoelectric point of that amino acid, then you have an overall negatively charged amino acid, okay? Now, why is that important? Why do we care? All right. Well, as we said before, you know, at the isoelectric point, it exists as a neutral dipolar Zwitter-ion. But if you can control what kind of charge is on that amino acid by lowering the pH or increasing the pH above or below that isoelectronic point, then what you can do is you can exploit that to separate out not only amino acids, but things made of amino acids like peptides, like proteins. And so this is used quite a bit to separate out mixtures of amino acids and proteins. They just manipulate the pH, and then they apply a voltage essentially across the solution. Usually it's on a gel, like agarose gel or page. I remember the acronym PAGE, page gel. I've done this, but it's been decades ago that I did this. But and you can use that gel as sort of a template to separate out the different amino acids or proteins, or based on the pH and the isoelectric points of the different compounds. Okay, but that's the extent of it. We're not going to really, I can ask you some conceptual kind of questions about this, but we're not going to do anything real strict and challenging with this. Okay. So again, now that we know about the amide functional group and we looked into that a little bit more depth, we can say that, well, this is how we connect amino acids together. The nitrogen acts as the nucleophile and attacks the carboxylic acid. We do a condensation reaction where we spit out a water molecule and we create that amide functional group. Okay, so you can see that in these molecules, you got the C double bond O and it's connected to the nitrogen. Okay, that the nitrogen in this case is going to be a second. It has one other substituent on it, still one hydrogen, but there is one amino acid that where it would not be a hydrogen here. It would be another R group, and that's proline. Okay, but again, you don't need to memorize those, but recognize that not all of them have an NH bond. All right, but the other thing that is important about that amide functional group, remember, is that you can draw different resonance forms for that. One of them, where the lone pair of the nitrogen gets pushed down to become a double bond between the nitrogen and the carbon, which would put a formal positive charge on the nitrogen. And at the same time, a pair of electrons move out of the pi bond between carbon and oxygen, moves up onto the oxygen to give the oxygen a negative charge. And so because of that delocalization, it means that all those atoms are going to be in the same plane. Right? That is a planar part of the molecule. And so that forms like the backbone of your protein. Okay? There's a restricted rotation around that carbon-nitrogen bond because of that. And so it kind of locks everything in place there. All right. So more terminology that we said that, you take the amino acids and you use them as building blocks for proteins. The correct word for that is residue. So the individual amino acid monomer type units are called residues. Okay. And as I said before, if you connect those together, that sequence gives us this long continuous chain of those amide functional group bonds. And that's what we call the protein's backbone. Okay, but in proteins, you'll also see this. And we talked about this before too. When we talked about thiols, you can produce these disulfide linkages, but it's only with the cysteine, and cysteine is another one of those amino acids. So when I say you don't have to memorize the 20 amino acids, you don't need to memorize the structure of cysteine, but two things about cysteine, you do need to remember about cysteine for quizzing and testing purposes, is that it's got sulfur in it, and that it can form, if two cysteines are in a good position relative to each other, they can form that disulfide linkage. All right, and you got the picture there in your handout as well. Basically, you oxidize these two thiol functional groups and you get the disulfide bond. Okay. And I think I mentioned this before that, essentially when you're styling your hair with heat, like a hairdryer or curling iron, you're sort of breaking these disulfide bonds, then the hair gets in whatever shape you want it to, and then they sort of reform into that shape. That's part of what. Of course, you also have gel and other, products. Not me, but of course. Anyway, I don't know why you like to use that analogy. Anyway, okay, let's move on. Okay, so speaking of proteins, we're going to go into some more details about the different types of proteins now. Their shape overall is sort of one of two. They can either be what we call fibrous or globular. And roughly speaking, fibrous proteins are, as the name implies, they're like fibers, okay? They're just straight chains, sort of organized, aligned side by side in long filaments. So fibrous proteins are comprised of polypeptide chains arranged side by side in long filaments. And the consequence of that, because there's all those intermolecular forces involved there with each other, between the different polypeptide chains, they typically are not going to be soluble in water. That's one consequence of that. But also they're going to be pretty tough. Okay? So this is where you would have like a structural protein. Muscle, keratin, that kind of thing. Anything that needs to provide structure to a tissue. But then the other one, the globular, they're typically coiled, more spherical shaped. And because of that, they are typically soluble in water. And because they're soluble in water, they can move around. Okay? So they're capable of motion within cells. Okay? Now, again, I'd be surprised if you haven't seen this information before in a biology course somewhere, but with proteins, there's 4 levels of structure. So, first of all, there's the primary structure, and that's the actual sequence of amino acids. Okay? Like glycine, cysteine, leucine, whatever. The sequence of amino acids is the primary structure. Okay, well, whatever that sequence is, that's going to have consequences that go beyond that ordering of residues. And so the first way in which that has consequences is evident in the secondary structure. Okay, and so that's just this regular pattern that develops from that chain of residues. So the secondary structure of a protein describes how the segments of the peptide backbone orient in a regular pattern. We'll see what those are in just the next slide here. But again, I think you've seen all this material before. But then even beyond that, all those intermolecular forces work together to give us the tertiary structure, which that's like the entire shape of the protein. Okay, so like where are the hydrophilic regions of the protein? Where are the hydrophobic regions of the protein? Okay, and what intermolecular forces are kind of responsible for doing that? So the tertiary structure describes how the entire protein molecule coils into an overall 3-dimensional shape. And then finally, as I said, most proteins, to my knowledge, are not just one chain of polypeptide, a bunch of amino acids connected together. It's usually multiple of those, 2, 3, 4 of those coming together in a certain specific way to give the protein a function. Okay, and that's what's reflected in the tertiary structure. So the quaternary structure. Quaternary structure describes how different protein molecules come together to yield large aggregate structures. I mentioned earlier that hemoglobin, the reason why it works the way it works is because it's 4 different proteins come together and interact in a very specific way. Okay. So the 2 secondary structures, we're going to kind of go through these. We already talked about the primary structure some, but we'll go through the others a little bit. So the 2 secondary structures are the alpha helix, and you can see that it's in your handouts. It's a little bit of a different diagram, but I did that on purpose because I want you to see that it's the hydrogen bonding that's typically responsible for this. Okay, this hydrogen bonding, like between this hydrogen that's connected to the nitrogen interacting with the nearby oxygen. So all of those working together in concert will create this alpha helix. Okay, that's one possible way they can organize themselves. So if you look at it with a ball and stick model, it'll look like this. But if a typical way of simplifying that are with these ribbons. Okay, so and you can see that the ribbon is in a spiral shape, an alpha helix. Okay, but the other way is the beta sheets. But to be more precise about it, it's like a pleated sheet. It's not a freshly ironed sheet. It's just pleated. So it's got that pattern of up and down, kind of wave tops structure. But again, it's just a different way to align all those molecules so that they can maximize the amount of hydrogen bonding, because that when you maximize the intermolecular forces, that lowers the energy, because those are attractive interactions. And so in this case, you can kind of see how they line up, the ribbons line up in sheets. Okay, so you just need to know that the two secondary structures are alpha helix, beta sheet, and know somewhat what they look like. Nothing specific, just, you know, coils versus like a sheet. Okay, all right. All right, so moving on to the tertiary protein structure. So as I said, this is, you put all of the intermolecular forces together, and what kind of shape does that produce in 3 dimensions? Well, roughly speaking, it's going to be highly influenced by 2 properties. Hydrophilic parts of the molecule will want to point outside where the water is. Remember, these are in, you know, living things. They're about 70% water. So the hydrophilic parts of the molecules that can interact favorably with water will point towards the water. Whereas the hydrophobic part of the molecules, like if it's got a lot of carbon in it, that's not going to want to interact with water at all. So it tends to get pointed to the interior of the molecule in the 3-dimensional shape of things. So hydrophilic interactions of the polar side chains versus the hydrophobic interactions of the non-polar side chains. That's really what's driving the tertiary structure of proteins. But also, we can't ignore that there's these disulfide bridges, which also will, you know, affect the overall shape as well. Okay, and as we have already said, the hydrogen bonding, because it's a strong intermolecular force, has already influenced the alpha helices and the beta sheets. And so just in, you know, the next order of magnitude will affect the 3-dimensional shape as well. So formation of hydrogen bonds between nearby amino acid residues. Okay, and one more that we haven't talked about is that, you know, depending upon if some of those oxygen have their negative charge, right? Like in the Zwitter ion, it does. So imagine like a calcium or a magnesium bridging 2 of those carboxylate groups. So we call those salt bridges. So like a metal ion that's got a +2 charge on it, like a magnesium or a calcium, can bring together 2 carboxylate groups, and that would affect the overall 3-dimensional structure, right? Because it would, you know, kind of lock those in place as a salt bridge. Okay. Now, tertiary structure is not something that's set in stone. It's something that depends on other conditions, like the temperature, like the pH. Those are the 2 big ones. And so whenever for example, whenever you cook meat, really what you're doing is you're breaking down the tertiary structure. But you could do it with other, you know, factors as well, like pH. So the tertiary structure of globular protein is the result of weak intermolecular attractions that can be disrupted by a change in temperature or pH, causing the protein to become denatured. Just know that term, denatured, and know what can cause it. Okay. All right. Okay, so we will talk a little bit more specifically about this type of protein, enzyme. Enzyme is a protein, but the reason why it gets more attention, it's you know, what we're going to focus on is because it is also a catalyst in biological systems. So it's a protein that behaves as a catalyst for a biological reaction. Now remember, a catalyst, what it does is it speeds up a reaction. Okay, it doesn't really allow you to produce something in a chemical reaction that would not otherwise be produced. But if it can speed up a certain chemical reaction, it might lead favorably to one product instead of another product. Remember, we talked about this before too, how there's kinetic control versus thermodynamic control. Well, enzymes help us to drive the kinetic control of things. Okay? How does it do that? Well, to any catalyst, the way it works is it lowers that activation energy. Remember that every chemical reaction has to get over some hump. We call it the activation energy. Well, a catalyst, through the mechanism of the reaction, just lowers that activation energy. So it increases the rate of reaction by decreasing the activation energy. Okay? The other interesting thing about enzymes is that most of them are very specific to a certain substrate. Will only facilitate this type of reaction in this type of molecule. Okay, but there are some that are more general, and the one that's listed there is papain. Okay, and that's a digestive enzyme, and so it basically will just, any protein, it just clips those amide linkages indiscriminately. Okay, but most catalysts are very specific for substrate and the way in which it transforms the substrate. Okay, so how does it do this? We look at a very, very simple model of how this is achieved. First of all, you've got to have this interaction between the enzyme and the substrate. You have to combine them. Okay, the substrate has to find the active site of the enzyme to form the enzyme substrate complex, E.S. And then, simply put, it converts the substrate into a product. So instead of E.S, we're going to indicate that with a E.P, P for product. Now that substrate could be, or pardon me, that conversion could be like adding a phosphate group or adding an acetyl group, something along those lines, facilitating decarboxylation maybe. There's all these different organic reactions, and we've gone over many of them, but I'll let you learn more specifically about them in biochemistry or molecular biology or whatever. Probably you get bits and pieces of it in different courses. But anyway, but the point being is that in this simple model, all we're concerned about is that it transforms that substrate into the product by facilitating, speeding up some specific chemical reaction. Okay, and then because the product is not, it's changed, it's been transformed. So now that molecule is not attracted to the active site. So it leaves. It gets released. So it releases the product from the complex, and you have the enzyme regenerated. So you got the enzyme back again, so it can take on another substrate and repeat the process over and over again. Okay, so this is just a diagram that illustrates this. So if we have an uncatalyzed reaction, we've got this pink line, or pink curve to indicate a pretty large activation energy. We'd have to get over that hump to create the product, to create this product. But with the enzyme catalyzed, it'll go through a different mechanism. It goes through maybe a couple steps. But overall, the activation energies are much lower. So it's much easier, it's faster to transform the substrate into the product that way. Okay? Now, the other thing that we want you to know about enzymes is that they oftentimes will require something else. So we call a cofactor, a non-protein component to facilitate the reaction, a cofactor. And it can come in different forms. Sometimes it's a metal ion like magnesium or zinc. We call those metalloenzymes. But other times it's an actual organic molecule or ion that's called a coenzyme. Okay, and you learn about these a lot again in your biology courses and whatnot. And they're typically derived from vitamins, so that's why it's so important to eat your vitamins, right? Okay. And so this isn't in your handout, but this is just a couple examples of cofactors. Okay, ATP isn't purely speaking a cofactor, but it provides, what it does is it provides energy to the enzyme so that it does kind of like a pinching action, it goes boom, and that's what facilitates the reaction. The energy release for when the ATP becomes ADP, it usually causes the enzyme to kind of clamp, and that facilitates whatever reaction it needs to take place. And then the coenzyme A, of course, is involved in glycolysis in other places. But just, you don't need to know that for this course, just giving you some examples here. Okay, and as I said, this is a very whittled down version of chapter 26. So, and when I looked at the end of chapter problems, these are the only two that I found that I would recommend. However, and I won't keep you, but I do have an active chemistry assignment with some more questions in it that I'll just make available. But you know, like I said, this is the last lesson, so I don't want to belabor it too much. But I'll open it up and let you guys use it to practice. And then remember on Friday, what we'll do on Friday is we'll do some practice problems together. And then we'll have our 11th and final pop quiz on Friday. So thanks for hanging in there, guys. I'll hang back if you got individual questions for me. Otherwise, have a great day.